Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Jason Coloma. Jason is the CEO of South San Francisco-based Maze Therapeutics. The company is developing small molecule drugs to halt or potentially reverse kidney diseases. The drug discovery work at Maze is grounded in human genetics. They look at large pools of data to see how variants of one kind or another can make people sick or protect them from falling ill. Maze went public in early 2025 after surviving several lean years of biotech financing. A key partnership unraveled after the federal government sought to block it on antitrust grounds. Maze found a way to bounce back and enticed a new partner to support that program. Those battles have strengthened the company's culture. Maze is now in position to find out in the next year or so whether or not its two lead programs, an APOL1 directed kidney drug and another aimed at SLC6A19, are good enough to pass phase 2 clinical trial scrutiny. Like many biotech industry leaders, Jason didn't take a linear path into this moment of possibility in medicine. And that's part of what makes his story and Maze's story so interesting. Now, before we get started, a word from the sponsor of the show, AlphaSense. So far, 2026 has been a year of transition for healthcare and life sciences. Over the first half of this year, the AlphaSense healthcare research team analyzed individual stories moving the needle across the life sciences ecosystem as part of the Sector Spotlight series. The 8 spotlights can be downloaded at timmermanreport.com. They feature the high-stakes battles for new drug markets like oral GLP-1s and targeted radioligand therapies, a resurgent biotech IPO market, and more. Follow the sector's key debates during the second half of 2026 and beyond with the latest sector spotlights directly in AlphaSense. And quick question. When was the last time a CRO told you what something costs before you signed an NDA, sat through 2 scoping calls, and waited a week for a scope of work? In bioanalysis, pricing gets treated like a closely guarded secret. Dash Bio thinks that's backward. It publishes pricing publicly on its website and even has a calculator that shows how the price changes if you redesign your study. You can see what your study costs before you talk to a human being. No surprise line items, no it depends, no games. Dash was built to be the bioanalysis company that's actually straightforward to work with. Transparent pricing, guaranteed timelines, and data in days, not months. If you're tired of pricing that feels like a negotiation every single time, run your bioanalysis with Dash. Visit dash.bio/pricing and see exactly how much your next bioanalysis study will cost. Now, please enjoy this conversation with Jason Coloma on The Long Run. Jason Coloma, welcome to The Long Run.
Yeah, thanks for having me, Luke. Appreciate it.
So, Jason, I'm eager to dive in on this story about Maze Therapeutics and how you got here, because this is one of those companies I covered from the very beginning. I don't know if you remember, but Series A, where you got cool science, prominent people, and a lot of money coming around the table around a big idea. That's kind of like the kind of thing I tend to focus on from early days, and I like to track over time. So this will be fun.
Yeah, no, thanks. That's great.
So let's start from the beginning with you. Where are you originally from?
Yeah, I mean, I was my family immigrated here from the Philippines back in the 60s, and they didn't come together. They actually met here. So I was born in the U.S., but, came from an immigrant family, and my dad was military. So we actually moved around a bit. Which was great. But at the same time, not easy as a kid to kind of always be sort of the outsider looking in, always adjusting to new environments. And then, but, what I call home is California. So I went to high school in Northern California, ended up going to school in the Bay Area, spent some time, on the East Coast and in Europe too. But, fundamentally, I still think of the Bay Area as my home.
So you did those high school years and college years in the Bay Area. Um, what did you said your dad was in the Army, or in the
Oh, he
military?
was Air Force, but yeah.
Air Force. Okay.
Yeah.
And what did your mom do?
She was a nurse. So she moved here. There were a lot of nurses who had moved, I think, during that era to America. She started out in the Midwest in Chicago. That was where she first started as a nurse, and then eventually made her way, um, to California, and that's where she met my dad.
Okay. Okay. And what kind of values did they seek to instill in you and your siblings, if you have some?
Yeah, no, I mean, I think, we were very, a middle class family, hardworking immigrant, trying to, how do you adjust to a new country? They both spoke English, which, helped. But, um, I think, just how do you adapt to a new environment, trying to see how you can be resilient to that type of change. I mean, my mom came here pretty young. She was probably 20, 22 when she moved here. One of those stories of like having $20 in your pocket, kind of, and the clothes in your suitcase. And so, that they can't help but kind of pass on those kind of lessons, what it means to be an immigrant family, how to make your way in America, how to adjust and sort of adapt to a culture that wasn't yours, and calling it home. And compounded with the fact that we moved around a little bit, how always having to constantly adjust, and to be adaptable, it was sort of part of that feeling. I think a lot of immigrant families kind of have that sort of, how do you be resilient, that hardworking attitude, how do you adjust, and a certain level of resilience, which I think over time has obviously been very helpful for me as I kind of thought about that through the years.
Yeah. So the Bay Area became your adopted home. And what kind of schools did you attend there, or what things interested you in those kind of high school and undergraduate years?
Yeah, I was always interested. I always gravitated towards science. I also, and a little bit of like sort of what that meant. It was a little bit for my mom was a nurse, but my dad obviously had a very different background being in the military. So it wasn't that like hardcore basic science was, it was always sort of in the applied way. I think that's very common of immigrant families to kind of think about, should you go into medicine? Should you go into, be a pharmacist? I was, initially just very interested in basic science. So I had thought about, when I was younger, because of my dad's influence, thought about going to the military academy. I was accepted into the military academy, but realizing that wasn't really for me.
Which one? Was it Air Force at Colorado Springs?
Yeah, exactly. Yeah, it's just following sort of in that, following your father's footsteps, so to speak.
Yeah.
And it was a little bit of a hard conversation because I decided not to go. My dad was really intent on that. It became a pretty sore subject actually over time. During that time, sorry, we didn't actually talk after I decided not to go to the Air Force Academy. Very, very tense time of my life. And I was kind of on my own a little bit because, and so what ended up happening, I really didn't know where I was going to go to school because I had turned down the schools by then, and including the academy. And so, but fortunately, one of my counselors in high school, took me under their wing and, so encouraged me to apply for a scholarship at University of San Francisco, which is a Jesuit school. So I came through more of a Catholic Jesuit kind of undergrad training. Fortunately, I was able to go there. Sort of had that type of training from the Jesuits, which really still sort of is a reflection of me and sort of like the way that I kind of think about the world. Um, and then
Yeah,
eventually,
for those who aren't that familiar, Jason, the Jesuits are a branch of the Catholic Church that's very focused on public service, like service to others. Would you say, or what else would you say you picked up in terms of influences during
yeah, I
those
think,
years?
sir, that's a good point. Yeah, so I think the Jesuits really embodied academic rigor as well as a commitment to service, which I think I've carried through the years, Luke. Being able to do good and as well as They were at times, if you look through history, they were also a little bit of the more rebellious group within the Catholic Church,
Hmm.
uh, which kind of stuck with me as well. And I thought that was, really interesting to kind of, learn with that type of background, again, of academic rigor, as well as sort of like this commitment to service in the community, which was embodied in sort of like the way that, we would do things in San Francisco in particular, which I could talk about in a second. And then, really just kind of thinking holistically about the world that, you have this embodiment of what your belief system is, and your relationship with God, but at the same time, it's okay to have questions about that, which I think they encouraged, which was kind of different than the way that I was kind of raised and grew up.
Yeah. So were you on a science track in those years, or did you not really know what you wanted to do?
No, so I always was really interested in basic, sort of science. So I did study science, molecular biology, and kind of went through that kind of training, and really sort of like that. And so, as I was kind of saying, usually, immigrant families, they kind of like encourage people to go more applied sort of, medical school, pharmacy, dental school. I wasn't really interested in that. That's what ended up more in a research field, which is why I ended up at Berkeley. Really kind of developing an interest. At the time, I was interested in infectious diseases, partly because of this sort of connection we were having with the Jesuits and the service. Public health was actually where I was, interested in applying some of those basic research. And I studied not only infectious diseases, immunology, but also epidemiology at the time when I made that transition to grad school. Um, and then, kind of that started my sort of like more graduate type of training, which eventually I made my way into industry. But that was, at that time, I was really focused on really just being able to apply that more from a public health setting.
Who were some of your key influences, either at USF or during graduate school at Berkeley?
Yeah, I mean, there was a lot of people. Of course, I mean, undergrad, Mary Jane Niles was great. She was an immunology professor there, and she had trained at Berkeley, and so there was a natural sort of connection there. When I was at Berkeley in the public health kind of arena, there was George Sensabaugh, who actually was a chair of the department for a bit, who really talked about sort of like this, how do you apply this and make the largest impact to human health, which was always sort of encouraging to kind of think about, which eventually led my way into, thinking about developing medicines because I thought, how do you think about, large scale impact to health? And I thought, those kind of professors really sort of got me to think about that. That wasn't like a natural path for me. I didn't really know what that was, just given my background, who my parents were, and so eventually kind of making my way there of how do I make the biggest impact to human health, but not being a medical doctor, right? And that's what kind of eventually led me into I can make the biggest impact by helping and trying to advance medicine and trying to make medicines for patients. That ultimately is where it landed.
So you were not thinking about getting on the classic academic track. You were thinking about industry from, somewhere along the way during graduate school.
Yeah, I ended up kind of transitioning to a biotech. At that time, it's like late 90s, early 2000s. It wasn't a big, there wasn't a large, like what it is today, lots of companies forming, lots of venture capital. It was a very different kind of venture capital was starting to, emerge. Biotech IPOs was a pretty foreign thing. But I ended up at a company that still exists today, Cytokinetics. So I ended up at
Oh yeah.
Cytokinetics, which, know many of the people there. Some of the people that here at Mays actually also were from Cytokinetics. Um, and it was great. It was a different company, of course. It wasn't just focused on cardiovascular disease like it is today, or muscle biology as they are. We were a little bit more diverse, lots of different research areas and a lot of different therapeutic areas. And, transitioning from, more of an academic training to a industry training was great. It was the first time kind of working at a biotech, what that meant. Um, Kind of being exposed to the business elements of things, but not really, because you're still in research. Um, and then kind of, we had the fortune at the time to go public. It seems so long ago, of course, but it was a big deal because not that many biotechs were going public. I remember I had to look it up, but the offering was like 75 million bucks, but it was such a big deal
But
at the
you were a
time.
junior guy, like a staff scientist at the time?
Yeah, I was just working on, and we were working on, trying to think about different sort of platform technologies to better understand. I guess they would call that like phenotypic screening now, which some companies are using. But we were trying to use different elements of that to better understand the cell and cellular biology. And so that was it was great. And my one of my hiring managers, and it's a little bit of a story of you never know who you're going to run to later in life. His name is Jeff Feiner. He's the CEO of Septerna. We were
Yeah.
at Rock together. And yeah, no, I mean, it was many years later that I got reunited with Jeff, and we ended up working on a concept that became Maze, and it is I love that in the sense of yeah, you never know. You're just starting off earlier in your career, the connections that you make, and eventually either intentionally or unintentionally kind of foster they can benefit. I try
It's
to
a
tell.
good it's a
Yeah.
good lesson for a lot of young people to just find an environment where, you have a good boss, there are people working on cool stuff, and there's a chance to grow. And it sounds like you found that right out of graduate school at Cytokinetics. And for, added bonus, I mean, it's a company, so presumably they pay you a fairly decent salary and benefits. And mom and dad will let you come home for Christmas.
Oh yeah, it was great. I was like, well, oh wow, the sodas are free and their snacks are like, this is great. Coming obviously, from a grad school postdoc kind of level to being in industry, it was a lot of learnings. And yeah, I think that was the first place I started to like, oh, there's a business side to this, right? Which I didn't really think about. And there were people that, had different type of functions. James Sabry was our first CEO, but then we had Robert Blum, who was still the current CEO, kind of transition. James was more of a clinician scientist. Robert was more of obviously more commercial slash finance. But both of them were great. Many people still at the company, like Robert, who have, shepherded them to what it is today. So, and then we actually even, when I fast forward into Maze, we even had people coming here that I worked with all those years ago. So it was really nice to be able to make those connections. But wonderful time when being at your first exposure to like sort of the business side of things, which things like business development, which is a pretty common term now. And at the time I was like, what is that? Like, going to the business development people and going to Robert, like, what do you do all day? What is, what does it mean to do business? But obviously they had struck partnerships and how that adds value to the business and the company, the risk. But, being a scientist at the time, like not knowing that language, that vocabulary, right, was, which eventually led me to get an MBA, and kind of honed that a little bit. But yeah, it was my first exposure to it. It was great.
Well, what was it about the business side that intrigued you and maybe made you think that it suited you and your skills?
Yeah, it was. I think part of it was what I noticed, at least when people that kind of really, even if they didn't have a PhD or an MD, and can really understand and really drive, the company to move forward. They were really good at like integrating this concept, Luke, of like science, medicine, and business. And they kind of appreciated the depths of all of that. I think of people like at Third Rock who kind of mentor like Alexis Borisy, other people like that that kind of helped me kind of think about company building. And over time, I mean, to be that deep and articulate on the science and the medicine and the business, that's what started to get me interested. There were people like that, like including Robert, of course, at Cytokinetics, who had that sort of element to them to really appreciate that feature. And that started to get interesting to me, right? Like, how do you build a company like Cyto? How do you make it successful? How do you keep the lights on even in dark times? I think all of those kind of principles, were kind of interesting to me. And then obviously trying to then learn the vocabulary and the methodologies of what's important more on the financial business side was a clear gap to me, which I ended up, going back to school for.
How did you end up going? Because I guess you went to Dartmouth for your MBA, so you moved across the country.
Yeah, come on.
And I mean, did they have a biotech program
No,
specifically? Because I don't think many places do.
no. I mean, you would think of more Wharton. You would think of, even, there was an individual there. So I was, applying to probably the usual places one would apply, like a Wharton, which I actually got in at the time. But there was the guy there who's great, mentor at Cytokinetics, Jay Troutman, who actually was running the platform team. And I told him I was thinking about doing this, and Jay was like, "Have you ever thought about here?" I was like, "What? No, right? That's like." It's in the middle of nowhere. It's cold. Why would I? We said, no. I mean, why don't you? And he was like, why don't you meet a couple of alums? He had some nice interactions with some alumni. He's like, that is a very strong alumni group, even if it's smaller. You might like the experience just because it's so different since you've been primarily a Northern California guy. And if you're there for a couple of years, you might build, relationships and Jay, Jay Troutman was right. It was great. It was a completely different experience, right? Going over to the Northeast, they have snow, above your head, piles of snow, most of the year. This is the first time I realized spring doesn't actually start in March, like it does in California. But it was tremendous. It was great. Like understanding it was a small community. It's a very small, it's a smaller MBA program, but they can really focus on the students. Some of the, my best friends have come through there. I'm on the board of Alloy Therapeutics for a while. Was a classmate, Eric Anderson, who actually was one of the first guys at Adamab, which is very successful, protein engineering company. So it's a small, but I think very close group. And it was a very just different experience than I've ever had before, which was being in the Northeast, being in a very cold environment. I remember, like going to my car in the morning, Luke, and I never lived in an environment like that. And you turn on the car and the thermometer is like 0. Like, is this thing broken? Like, oh no, it's 0. It's probably even colder than 0,
But were you thinking then that you were getting this degree to help you advance in biotech? I mean, that you'd have a PhD and
Yeah.
BA, and that's so that you really could live across both worlds of science and business and build companies.
Yeah, absolutely. I think that was sort of like I went into that, and I think it was a different era. People say, would you have gotten an MBA like in this kind of environment? I think it was a different time where, to make that transition from, okay, I'm going to be more on the science side to the business side. Now it's pretty natural, right? Like if you look around, people go straight to Third Rock or they go right into a role from graduate school. The MBA is a little bit more optional because you can kind of develop those skills in other ways. This is like early 2000s, right? So the idea of like transitioning to that was not as natural. There weren't as many, opportunities, and there was a much smaller, I would just say, ecosystem of startups, right? Third Rock didn't exist yet at this period,
Yeah.
right? It wasn't a, firm of like, a company creating firms that were creating all these startups and all this venture capital, much different era. So for me to make that kind of like, if I knew I wanted to try to do something a little bit more business oriented, this was probably the best way for me to do it. And it worked out.
So after you got your MBA, was it then that you moved back to California for Genentech?
Yeah, so I ended up more on the business side, which was interesting, but I wasn't I put more of the Roche experience on my, because it was more formative in the sense that we got bought pretty quick. By the time I guess I graduated the program 7, we would, '09 was when Genentech I guess Roche had bought the rest of Genentech.
Yes.
And then I remember, sort of going, oh, wow, okay, what is this? Because Genentech was a wonderful, and it is a wonderful place to learn and work. But it was very U.S. focused, right? We only had, U.S. commercial rights, and it was a very U.S. focused kind of, I felt, culture, right? What was really more transformative for me was after the acquisition, I actually moved to the headquarters and worked in Basel, and that was really my kind of first experience obviously working outside the US, but really being exposed to, even if you think about it, cytokinetics, small company, Genentech, big, but very US focused. Roche was the first time I really felt learning on an international level what it meant to develop medicines for patients.
Well,
Different.
not only was that international experience, but this was, I mean, the biggest merger ever in biotech. And I remember covering this. There was all this concern about will the Genentech culture, that freewheeling, irreverent, scientifically creative
Yeah.
culture that made Genentech great, will that survive inside the larger company, the Swiss, more conservative or
No.
buttoned down, or or will they mess it up somehow? And so you would have been right there inside when everybody was trying to make it work.
Yeah, it was a very interesting time to see that. It was, I would say, Roche did a really commendable job to try, because what they were also buying it because the Roche pipeline, admittedly, was not advancing in a way that they wanted to, and really innovated. There was a reason, of course, and they wanted to instill some of that culture that you just talked about, Luke. And how do you, sort of not only think about, sort of the best science, but also how do you create a culture that can be that creative and think differently and be a little bit brazen, kind of in the way. So it was interesting. It felt like a reverse of that. I think that's why they moved so many of us to Basel, is to actually try to cross pollinate this idea that we really can bring some of that culture to a very traditional Swiss culture. When I first moved there, because it was pretty early in that, it was probably towards the end of 11, 12, 2012 or 12, 2012. And yeah, when I first got there, Basel was still very formal. People will still walk around in suits and ties, and sort of be very, very formal in their interactions. And you can imagine a bunch of Californians coming in blue jeans and t-shirts, going to meetings. It was a little bit of a sort of awkwardness in this, and it was interesting because over my time there, which was about 5 years. A little over 5 years, sorry. Um, it really changed. It was kind of awkward at first because you see, Swiss German folks and very European folks trying to dress more casual. They would wear jeans, but they would be ironed, for example. Still wearing a blazer to try to look very, sort of. They started to try to relax that, and more leadership roles were, of course, moving to people from Genentech and so forth. But it was really interesting to see that they were Roche themselves were committed to doing that and trying to learn and be able to try to as best as they could, right? Because of course people are going to leave, and people ended up moving on to do great things. But they tried their best to retain the culture as long as they could, which I thought was very redeeming.
Would you say that the merger was successful looking back? I mean, I know people have looked at this, and it's been a while since I've looked at it, but in terms of the pipeline productivity and, however you want to measure it, did it work?
Yeah, I was going to say, it depends how you think about what would be success criteria or factors in there. I mean, I think in terms of trying to make Roche into one, yes, at the time they really did, improve their pipeline, both the European part and through the innovation that was coming through Genentech and Chugai as part of that R&D engine, which was a Japanese affiliate. Yeah, I would say on that front, yes, very much so. More innovative, more you can even see that with some of the people they've brought in for leadership roles now, like Aviv Regev running R&D for Genentech. I think they went very much were able to do that. I think it's hard to pipeline productivity. Yeah, it was interesting to see that transformation. This was the era where Roche and Genentech lost a little bit of the luster around. They were the market leader in oncology, right?
Yeah.
But obviously that changed quite dramatically with the advent of immuno-oncology. And, they unfortunately weren't able to do that, but they transformed the pipeline in a different way, which who would have thought, their three biggest sellers are MS, hemophilia, and an ophthalmology drug, right?
Yeah.
And now, making a huge bet into cardio metabolism. So, yeah, I think that's what I think in general you would look at that as obviously success on multiple fronts, and I think they're still trying to figure out because they seem to really want to be committed to this a multi-prong R&D engine with three different R&D units. So, which they still have.
Well, so you were there, you got this international experience, you got kind of the an inside look into how big companies operate, but also how you maintain that creative startup vibe, if you want to call it, at
Yeah,
Genent. So you got a really good experience. How did you end up going to Third Rock?
yeah. I was in business development by this period in the partnering group. Sophie Konorski was the boss at the time. And the CEO, who was Severin Schwan, was interested in sort of what brought together the two divisions, which was the diagnostics division and the pharma division. And the belief was there were certain things, including data, that can kind of merge those things together. And so, I've primarily been doing, typical pharma type deals, products and, technologies and, academic partnerships. And for whatever reason, Sophie gave me this project, which was to help the executive committee kind of think about, oh, what is the thing that ties us together? Which led us eventually into the strategy around personalized healthcare that was looking at things like Flatiron, foundation medicine, these kind of things that could be, data and technologies that can, if you will, bring together the diagnostics division and the pharma division to enable not only identifying patients, but treat them better. So those deals came out of a strategy that I was able to help architect with Sophie and Dan O'Day at the time was the head of pharma. For whatever reason, they gave me that project, Luke. But that gave me the opportunity to not only think about different types of deal structures, think about precision medicine in a different way, think about the importance of computational capabilities and data as it applies to biology and drug development. And then it also, which was your real question, was start to interact with people at Third Rock. So Foundation Medicine was founded by Third Rock. Alexis Borsi was, one of the founding members before the management team took over. Neil Exter, who was a partner there at Third Rock. They had a model at Third Rock, which they still do, which is form the ideas, form the companies as venture capitalists, but then, we run those companies for a while and then eventually bring in the management team. And it was really about this idea that led, to Foundation Medicine. And so that was the first time I met some of them.
Now,
We
for
did.
folks who really aren't familiar, Flatiron Health, Foundation Medicine, those were a couple companies that were acquired by Roche, I think in pretty short order around this strategy of, I don't know how molecular diagnostics tied to therapeutic development. So in other words, if you better understand the target you're going after, like say HER2 for Herceptin, you really ought to improve the probability of success in clinical development. Better understanding of biology, speed up, improve the success of your clinical trials, come up with great new therapies.
Yeah. No, better said than I did.
But you lived it. You worked on this, and so you got to know Third Rock.
Yeah.
And how did this end up becoming a job for you?
Yeah. So I start, and then I think as kind of those conversations, I think it was, you start building, I think the way that now that I know this, venture capitalists kind of build these relationships with folks. And as you can imagine, there's no real job board for venture. That's not like they post like, hey, well, I guess they do now, but at the time they don't really say, hey, we're hiring. It becomes a little bit more organic in sort of relationships and individuals. And so I got to know the Third Rock folks. I had worked on a couple other company partnerships with them, including Blueprint Medicines, one of the first partnerships when Christoph Langauer was the CSO there. And Jeff Albers was the CEO. So, again, sort of like making, sort of more connections within the Third Rock ecosystem. And then over time, just building those relationships, they finally said, "Hey, have you ever thought about either, coming into a company or coming to work here?" At the time, they were saying like it had to be in Boston. Third Rock is primarily, based in Boston. They didn't really have a San Francisco office at the time, or a small one. And I said no. So I said, I'm not going to live in Boston. I already did my time there through in the Northeast through Dartmouth. I'd rather go home. I came back to California. And then I got as probably Alexis Borges want to do. I get a random text from Alexis and said, "Hey, let's go grab lunch. I'm in town." And then I guess in stereotypical fashion, we met at the Rosewood Hotel and so, on Sand Hill Road. Alexis took me to lunch and he kind of articulated, "Hey, I think, we're going to try to actually in this fund, we're going to try to kind of build a little bit more in San Francisco. Would you like to be part of it?" And I was like, wow, okay. And it was great. I mean, I transitioned over to Third Rock. I had been, primarily by this time, more of a big company guy, Luke, right? Just more big pharma perspective. I kind of describe this period at Third Rock where I got to work on some pretty cool things with Alexis and the others. It's kind of like a postdoc in entrepreneurship, to be honest. It was pretty cool to be, you had people like that, just, Charles Hompsey, Mark, the founder, Kevin Starr was around a little bit. And so it was nice to just be able to pick their brains and, what they can learn. It was a little bit of a safer environment to learn entrepreneurship, to be honest, than just going straight into the company. So it was a real blessing to be able to have that period to kind of understand how they were building these great companies that eventually became things like Revolution Medicine.
So far, 2026 has been a year of transition for healthcare and life sciences. Over the first half of the year, the AlphaSense healthcare research team analyzed the individual stories moving the needle across the life sciences ecosystem as part of the Sector Spotlight series. The 8 spotlights can be downloaded at timmermanreport.com. They feature the high-stakes battles for new drug markets, like oral GLP-1s and targeted radioligand therapies, a resurgent biotech IPO market, and more. Follow the sector's key debates during the second half of 2026 and beyond with the latest sector spotlights directly in AlphaSense. And quick question, when was the last time a CRO told you what something costs before you signed an NDA, sat through 2 scoping calls, and waited a week for a scope of work? In bioanalysis, pricing gets treated like a closely guarded secret. Dash Bio thinks that's backward. It publishes pricing publicly on its website and even has a calculator that shows how the price changes if you redesign your study. You can see what your study costs before you talk to a human being. No surprise line items, no it depends, no games. Dash was built to be the bioanalysis company that's actually straightforward to work with. Transparent pricing, guaranteed timelines, and data in days, not months. If you're tired of pricing that feels like a negotiation every single time, run your bioanalysis with Dash. Visit dash.bio/pricing and see exactly how much your next bioanalysis study will cost. You were, I guess, a venture partner or an associate or something like, within this company creation firm. They have projects that are kind of like proto companies. They're trying to figure out if it will become a company. And were these the kind of things that you got to work on?
Yeah, so I started off as an entrepreneur in residence, got moved quickly into a venture partner role, which is supporting the partners on how to think about different ideas, and you see if they beat them up if they had a particular process they like to go through to build up, and whether there's a real investment thesis around a company, and then eventually build those companies and run them for a short period. So it was really cool. You got a little bit of the whole sort of taking an idea trying to formulate it into something that could be a company, actually help build the first stages of that company, and then actually have a fractional management position at the company to help stand it up. And then, because I had worked at Roche, and I was really interested in, I had done the Flatiron deal, and, kind of did the, foundation medicine concept. I was interested in this convergence concept of, data, computational biology meets drug discovery. We weren't calling it, machine learning AI yet. But the idea of like this convergence was interesting. It was interesting to folks and the partnership at the time, like Christoph and Alexis. And so we started to build out concepts like that. So I got to work on a few things while I was there. We worked on Celsius Therapeutics, which was based in Boston, which was working with a few founders, including Aviv Regev. She was, one of the things that had kind of spun out of the Broad. That eventually got bought by AbbVie. But that concept of how do you use single cell genomics to really guide a better understanding of biology, such that you can develop better therapies for patients. That was what Celsius Therapeutics was before it got bought. Got to work a little bit on what eventually, with Alexis and Christoph, what eventually became Thrive, which was a diagnostics kind of company to really think about using sequencing and other ways to better diagnose cancer patients. Thrive was eventually bought by Exact, which Exact eventually got bought by Abbott. So that was really fun to kind of work on, sort of taking that concept into a company.
This idea that you can screen for 50 or so different types of cancer on a simple blood draw,
Yeah.
with high-powered, fast, cheap DNA sequencing.
Which was, sounds like a little obvious now that people should be thinking about this, but at the time, it was Bert Vogelstein, who is a preeminent, researcher out of Johns Hopkins. Christoph Lange and I were actually trained in that lab, so I had a really good relationship with Bert, and he had developed this concept with some of his people there, but it wasn't really taking off as a business, and because some of us, and we ended up bringing some of the like Steve Kafka in from Foundation Medicine to kind of help really take this concept and turn it into an actual company, was great. It was actually really neat to see all of this starting to come together from the standpoint of things that we had kind of worked on in the past, and can kind of help a technology and a founder like Bert to really advance that. And then look where it is now. It's actually sitting Exact, taking that to the next level, and hopefully Abbott doing the same thing. So that was really cool to see. And then I also helped out Daphne Kohler at In Citro, which was, sort of like one of the first machine learning on top of, looking at AI as she was trying to get off the ground, and trying to build out the sort of like research platform effort that eventually became In Citro. And then I started to work with Charles Homsey, who was one of the co-founders of Myocardia. He had just left the board of Myocardia, come back to Third Rock to kind of work on some ideas. And he wanted to, think about different, more genetically driven approaches, which ended up becoming Maze Therapeutics.
Yeah. So, by the way, Daphne Kohler's a previous guest on the Long Run podcast. I should go back and surface that one for folks who are curious about the origins of AI and drug discovery. This was probably 10 years ago by now. Or close to it. But so Maze, what was the big idea there? Your discussions with Charles Homsey, what excited you about this one?
Yeah, so Charles, so the concept, Charles has just come off Myocardia. He co-founded BridgeBio too with Neil Kumar, thinking about more monogenic diseases. And at the highest level, when he started to pitch me on sort of like helping him work on it was, and Jeff Feiner, who I mentioned earlier, was, let's, Bridge is kind of working on monogenic diseases. How do we think about more complex diseases? And are there ways to actually use genetics plus, what we're starting to see and emerging in the tools like, single cell genomics and other, perturbation techniques, to better understand, can we start to segment or better understand subpopulations of more complex diseases so that we can develop more medicines. That was like the highest level. And part of it was, you had these big data sets now, like UK Biobank. You also had FinGen, these large scale initiatives and within countries that had genetic with paired clinical information over a longitudinal basis, where if you can kind of have the right people analyzing that type of information and data, you might be able to glean certain insights that allow you to, target populations. So if you take, like, for example, chronic kidney disease, the 37 million people in the US have what they call chronic kidney disease, which means there's probably different ways to kind of like segment that out. And if we could do that in a more targeted fashion, we might be able to actually, help those patients. So that was like the biggest sort of like interesting kind of concept was, okay, how do you take more complex diseases, use the tools and the data that are starting to emerge, and then marry that with more classical drug hunting techniques in a way that we can, sort of treat diseases that haven't seen a lot of innovation before, like kidney disease.
And these data sets have traditionally been just very fragmented or incomplete. But you had been exposed with the beginnings of Flatiron and Foundation Medicine. This whole wave of data was coming, and it was beginning to populate these databases, like the ones you said, UK Biobank, FinGen, there are others. And you thought, okay, if people can connect the dots, and maybe that's you guys, can see relationships between okay, here's a gene perturbation, and it really does lead downstream. Maybe it takes some time. We look at the medical records, we can see, yep, they develop chronic kidney disease at a pretty high rate. That might be a new target for drug discovery.
Yeah, absolutely. Or we can also look at those same data sets and go, wow, these people these people should have been sick based off of that actual analysis. Do they have a variant of the same gene that is actually protecting them from disease? And if we can understand that function, perhaps that's a way that we can, if in our language, phenocopy or copy that protective effect with a therapy. And that was sort of the general principle that led us to work on things like APOL1 kidney disease, because that's exactly what we found. There's particular APOL1 kidney disease is a genetically defined kidney disease that disproportionately affects the Black community. For people interested, it's interesting because it was evolutionarily conserved to protect people from West Africa from this horrible thing, African sleeping sickness. The problem is if you live long enough, people ended up developing kidney disease. It's a little like sickle cell and malaria. So similar sort of, in one context, it protects you from an infectious disease, but then the negative complication, if you have two copies of the variant, you end up having disease. So that interested us. But there were also, what we also found in those data sets, which we published on with folks like Eric Green and others in academia, is that, there were people that they had, even though when they had two copies of the variant and they should have got sick, they didn't. So that was interesting to us. And they ended up having a different variant that protected them. And we published on that. So if we could, and that was the thought, is if we can phenocopy that with a small molecule, well, we might be able to help some people. Right?
Yeah. And you were thinking about, everything that you were doing was going to be rooted in human genetics or guided by it.
Guided by, yeah, exactly.
You could say. And then you, what was your thinking that you would develop small molecules orally available, that was going to be the preferred modality, and then run them through the preclinical experiments and everything you've got to do to test the hypothesis?
Yeah. So that's where it starts to merge with like, well, these are interesting insights that one can glean, but what are you going to do with it? And then start to marry some of the more classical drug discovery. We had people from Cyto, and our head of drug discovery, had worked, was the head of drug discovery at Cytokinetics all those years ago. So we started getting some of those type of folks into the company. We actually started a little bit more broader with Charles. So in our model of Third Rock, Charles was the first CEO of the company. He had it a little bit more diverse in modality. looking at different things like antisense and other sort of ways to hit the targets. And then, eventually when I became the CEO, I felt, it's hard for a smaller company to do that. I know Rich Bio has been very successful at that, but I felt the right thing for Maze was to really focus on a modality because we had been building up all this biology expertise and genetics. That's probably good to simplify the modalities. And, so we focused on small molecules or pills to really kind of be the emphasis. And we had a really good set of drug hunters here who had worked on things like, from the companies I mentioned earlier that, had gotten things not only to the clinic, but more importantly to patients in the market. So since we were building that small molecule muscle internally, I really wanted to focus the company there.
Okay, so if you were taking biology risk on, say, novel targets, you could reduce your modality risk in the chemistry.
Yeah, I think that's the way to, you don't want to, I guess in business talk, you don't want to compound risk, novel biology with novel modality. Probably not a good recipe for value creation. That works out sometimes, and that's great for people. But, given the fact that we were looking at some more nascent developing stories like APOL1 kidney disease, I thought the best thing would be to, sort of like tried and true modality that we can match to that, and then really start to focus the company. We had a few other therapeutic areas at the time when Charles was the CEO, and I really wanted to focus the company on this question about complex disease of kidney disease in particular, because I felt like we could get better leverage in terms of expertise around a disease area. And really being able to focus the company, and it paid off. I mean, the markets have had some level of recovery, of course, recently, but there were some lean times during those years when we started the company 7 years ago. And the focus, paid off in the sense of being able to, be resilient during some pretty tough, capital raising periods.
Yeah, so the company actually got started with quite a lot. I think it was $190 million.
Yeah.
Charles was there at Series A. And then you came in pretty early. How big was the company? How far along was it when you took over as CEO?
Yeah, we were, I came in probably a year after the first funding. Um, and so that's in 2019. Yeah, we were probably still below 20 people. We had we had this small, it was actually down the street from where RevMed was, the smaller lab. That's where we got started. And then we moved over eventually to South San Francisco, here at Oyster Point Cove. So there were just a handful of us. We were sharing space with another Third Rock company at the time, just to kind of, defer costs and all that kind of stuff, share space. So we were probably, 20, 25 people when I took over.
And did you have all the same programs on the drawing board, like the APOL1, the SLC 6A19, and the others?
Yeah, in the business plan that we developed for Third Rock and the other investors, yeah, APOL1 was on there, SLC was on there. I look back a few months ago, and yeah, all of those were on there. We had GIS1, APOL1, and SLC, which are all still advancing in the clinic. So it was really nice to see that. We did have a couple neuro targets that the early team really liked. We ended up either partnering those programs or actually one of them is now our former chief scientific officer Eric Green is now the CEO of Trace, which is working in ALS. He took that program, and so we were able to at least advance it that way. But the core pipeline was there.
You had more programs than you could really develop on your own.
Yeah,
I mean, it's
Charles definitely had a lot of ideas. Yeah, Charles had a lot of ideas. It was actually a blessing to have that. But then it was more okay, how do we focus the company so we can build leverage and acknowledging that we're going to have to prioritize things, as we did raise a lot of money, but drug development, of course, is not cheap. So we just had to think of different ways to focus the pipeline, build leverage, and actually get these things into and through the clinic.
So you had a core focus on kidney became apparent with time. Um, these were all preclinical programs, I guess, still when you came
Yeah,
in.
they were all homegrown. We made
Yeah.
all of them.
And now, for what you had the Pompeii program, which is a little different.
Mhm.
I do want to ask you about this because this actually, while you were still a private company, became newsworthy in a way that I'm sure you didn't really love.
Yeah.
On how you struck this deal with Sanofi
Yep.
to co-develop this small molecule for Pompe disease.
Yep.
Sanofi, of course, was already the market leader with
Yep.
the injectable treatment for Pompe. And the Federal Trade Commission took a look at this partnership and said, "We don't like this. This is anti-competitive." And they basically forced Sanofi to walk away. Could you? I mean, this is.
Yeah.
There were a lot of people in the biotech community who were pretty upset about this form of government intervention at the time.
Yeah.
What happened there? And now that you've got some distance from it and you found another partner and it's moving along again, I guess, all's well that ends well, but what was that like?
Oh yeah, it was horrible. Because at this time, I made a decision, hey, I really want to focus on these programs, which, led to, okay, what are we going to do with the rest of the pipeline? Because we are making progress, but how do we advance them? And, we worked on a pretty hard target. Guess one, we knew a lot of other companies actually tried to do this and they'd failed for various chemistry reasons. And we actually made a nice breakthrough. And then in Pompeii, like you just pointed out, smaller therapeutic area in the sense of prevalence and incidence of the disease. And Sanofi was the leader. They were the most logical partner, and then they were excited about this. It was a target they had worked on. They weren't able to get through the chemistry effort themselves, and we figured out how to do it. And they wanted to do the partnership. We signed the partnership, and then of course, everything that you can read in the news. It was hard. It was a hard time because this was also a period where it wasn't that easy to raise capital. It was not the public equity markets that it is today, or the fundraising environment. And it was a pretty dark time. Trying to figure out. You kind of established a strategy. You're developing this plan. You've articulated this to the board because it was actually unusual. This was our lead program, Luke, right? And usually boards are like, "What are you doing? You want to partner the lead program? That's interesting." And but convinced the board this is the right thing, so we can focus on kidney. Got the deal done, we thought, and then this happens. And it was difficult for things that we couldn't really control, which was more a political topic, and then rather than anything. And so, unfortunately, of course, Sanofi backed down. We were fortunate enough to find a new partner, and yeah, I guess now with a few years of distance, I think for me, it's always it was interesting to there's so much work that goes into the program itself, but then when you're changing strategy, trying to get your board and your investors okay with that, and then more importantly, is just thinking about how do you have contingency plans, even though the best laid plans are not going to be able to be implemented because of things that you can't control. And so how do you think about not what going forward, everything that I talk about here and with the team is, yeah, I like this plan, but how do you think about a plan B? What do you think the alternative could be? How do we create different options so that we're not stuck in that period again that gave us so much uncertainty? I think that, to the degree that there are certain variables you might be able to control, that ends up, kind of being the lesson, at least for me. And I think we've been able to become, resilient. We were able to rally the employees around that. We found another partner thanks to our head of business development. His name's Atul. They did a wonderful job to find a great partner on Shinogi, pretty quick order. Um, and
It
then
was
we
only
were
like 6 months, I think.
yeah, I mean, everyone, because of my business development background, Luke, everyone's like, "Oh, wow, you must have figured that out." And I was like, "I know, credit where all the credit's due." Atul Dandekar, who's our chief strategy and business officer, had been working on this as a contingency plan, built a great relationship with the organization. And it's hard to do a deal that quickly. right?
You really needed to. I mean, you were burning
Yeah,
cash. You're a private company, and this was a
absolutely.
phase two asset, so the bills were starting to get bigger. I mean, right? And I doubt you
And I
could
have
go
a
back
plan
to
for
the
my
venture.
kidney. I have plans for kidney that's also burning capital, right? So
Right. And you're still, and you probably can only go back to the well so many times to the venture capital firms before, they want us,
absolutely.
they want this to be somebody else's investment so that they can exit. So you really had to do that deal.
Yeah, we had to. Unfortunately, we ended up doing a better deal than the one we signed with Sanofi, which is even a little bit more remarkable that Atul and the team were able to pull that off. Part of it is it does speak to we solved a problem that was really at the time, important for people to solve, and differentiated, and the quality of the data that came even from the early development studies, that I think people got comfortable paying that much for a phase one asset. But yeah, I mean, a lot of things had to go right. Atul and I and the team spent many, many hours late at night trying to figure out how we were going to get out of this. And fortunately, we did that. And it did help us kind of going forward, build a lot of resilience into the organization. The leadership team, obviously, you kind of bonded through sort of like an experience like that. And then it gave us a little bit of the confidence that whatever was kind of thrown at us, we can kind of figure out, you know?
You were battle tested
We were
and,
battle tested.
and ready to go public.
Yeah.
About a year later, again, when not a lot of companies were going public, it was a pretty
Yeah.
harsh environment. What was that experience like for you guys?
Yeah. I mean, again, yeah, you pointed out there was probably like no one was going public the year before. A couple of us tried to like see if there would be an opportunity. To your point, at some point, venture capitalists and early investors want to be able to see, find new investors, and we do too, in order to help grow the company to the next level. And so we waded through, it was a tough process. It honestly was, right? It was not in an environment that, was really conducive to what it is today. Now it's all about timing and context when you think about public markets. And it was not an easy process to get through. But I think, there was sort of like this quiet confidence that we were going to be able to get to the next point. Like what you just said, we felt battle tested and we've kind of figured, okay, this is the best way to get to the next step, even if it's not the optimal sort of like feeling when you're like going through that. But we were able to do it. And I think that was a testament to sort of the resiliency we've been able to build, and kind of over time just figuring out, okay, how do we continue to navigate these different environments? There's so many ups and downs to not only a company build, but also a medicine trying to get to, later stage development. But we had confidence in ourselves and in each other that we can get it done. And it was nice to actually see us pull it off.
Now, you talked a little bit about the APOL1 therapeutic hypothesis. That program is now in phase two.
Mm-hmm.
You've released a little bit of data on that. I know you expect more later this year and early 2027.
Yeah.
Can you say, what are your hopes? What's kind of the blue sky vision for that program?
Yeah, I think, APOL1 kidney disease, yeah, it's horrible in the sense there's no approved therapies. We know people, have faster progression to disease, which means they end up, what they call crashing into dialysis, which, once people are in dialysis, their outcomes aren't great. And as we talk to nephrologists and the patients and the parents of those patients, anything that you can do, because we know that standard of care or the current therapies available to them are just not working. They're not, people are still progressing. And so the idea is to try to develop, the first or one of the first disease modifying therapies such that patients can either elongate the time to, more end stage renal disease, or hopefully they never transition to that. Because we know if we can keep patients out of dialysis, Luke, we know that they just live longer, healthier lives. Once you're in dialysis, it's tough, not only in terms of outcomes, but it's also just tough livelihood. You talk to people that have to go do dialysis a couple of days a week, and It's not just that; it's like it's your family member or caregiver that has to do it. You're kind of wiped out the entire day of that because you just feel horrible. And then you know that it's really bad in terms of long-term outcomes for those patients. So anything that we can do that can sort of like be disease modifying that either delays or prevents that transition, that would be the goal here, because we know that the current battery of options, and nephrologists are saying after I use an SGLT2, that's it, or the last sort of like kind of therapy available. That's all I have. So if I have more options, this hopefully will keep people out of dialysis.
Now, for a long time, kidney disease development was, or drug development was, kind of stymied by the length. These clinical trials tend to be pretty long and
Right.
expensive, and like they have these hard outcome endpoints because the disease itself takes a long time to develop.
Hmm.
Um, Makes it hard to study. Now, but the FDA has been more open, I guess, in recent years in certain kidney diseases to allowing biomarkers in clinical
Yeah.
trials to give us an earlier read of efficacy. What have your interactions been like with the FDA on that count?
Yeah, I think, I mean, if you look at the innovation, prior to some of the recent, there was like a whole decade where you didn't have a new approval in kidney disease. The reason partially because, yeah, you took these long studies, outcome-based studies, which scare even pharma, because what that means is big studies with lots of patients and they take very long to do, which even dissuades even big companies like a Roche to not want to be able to develop in those therapeutic areas. But I think, companies in front of us, like Chinook and Travere, Vera, others, had started to kind of work with the agency and academic working groups working with the agency. And I think Eliza Thompson, who's the head of the cardio-renal division there, okay, well, are there things, if it's data driven on an indication by indication basis, can we look at different ways to actually be able to accelerate development pathways for drug if the data supports it? Right. So I think it's really nice. There's a disease, IgA nephropathy, which a lot of companies are participating and developing new therapies. There was nothing. Now there's a lot of different options for patients, which is great. Looking at different endpoints. Travere, which recently got an approval for a rare kidney disease called FSGS, off of just lowering the protein in the urine at all, which was a very dramatic shift in sort of an approvable endpoint. But a lot of that hard work, not just Travere, there was an academic working group called Parasol that was out of Michigan that really did engage the agency on that this was the right endpoint to really think about sort of an approval pathway for that indication. And then the cardiorenal division saying that, yeah, the data makes sense. Let's go for it. And Travere obviously recently, having an approval there for a disease that's been pretty horrible for patients. So it's really kind of neat to see that the FDA and this particular division is open to it. It is indication by indication basis and data driven. But the fact that they're willing to do that, and what's also nice to see is this collaboration of industry and academic working groups, plus groups like the nonprofit groups and patient advocacy groups like NEFCURE, public-private partnerships like the Kidney Health Initiative, all of these different groups that are really trying to figure out ways to productively work with the agency so we can see more medicines for kidney patients.
You're really outlining kind of the key ingredients for innovation to occur in a therapeutic area. I mean, you've got a good human genetic target, a therapeutic hypothesis. You've got a practical regulatory framework for demonstrating that something works. And it has attracted more academic interest, more companies, whether they be startups like some of the ones you mentioned. I know Vertex is also in the APOL1
Mhm.
area. It probably all, I mean, on one level you compete with these people, but you're also kind of showing each other the way forward and helping establish a path where anybody can be successful.
Yeah, it's an important point. I mean, investors who give us the money to allow us to develop these things, I mean, they want to have a good understanding of the regulatory pathway and the timing, and have some level of comfort that there is a a way to do that. And the more that that happens, the more, capital inflow goes into the therapeutic area. I don't think it's a surprise, after Chinook and others were kind of forging the way uh, when they were probably one of the first ones. But now, that's great. Those have shown that not only can make medicines, but investors can, sort of reap the benefits as well. That there's more companies forming in kidney, right? Like, and the number of companies that are forming is great. I think to your point about the academic community, it's interesting. The big conference in nephrology is the American Society of Nephrology, which will be in October this year. When we started going to that conference, Luke, it was pretty small. There weren't many industry people, mostly academic clinicians. Last year, you fast forward to sort of like ASN last year. I mean, almost a lot of pharmas, they actually had booths. That's when I guess a conference has arrived when you see the booths from the different companies. A lot more foot traffic. A lot more. When we first started, there were maybe a handful of publications and posters on April 1. There were probably like, I felt like, 8 times, 10 times more publications on the topic, so it's really kind of cool to see that when you're a little bit earlier, when no one's really paying attention to it, pharma industry, investors. It was funny because when I first took over for Charles as CEO, I started, I was doing a little bit of the which therapeutic areas should we focus on, and went to a couple of the investors who will remain nameless. They were like, "Oh, whatever you do, don't do kidney." I was like, "Really?" Oh,
For the reasons mentioned
the
earlier,
reason you
the
mentioned
time and
earlier.
expense, and
Time, expense, not a lot of approvals, regulatory pathways are, unclear. But I think we had done enough homework to realize that there were groups like that Michigan group, Parasol, there was NAFCure, there was the Kidney Health Initiative, there was a louder voice in Washington, and the administration and, the cardiorenal division really being open to innovation. So I'm glad we made that bet.
well, it's good to be early or ahead of the curve, but not too far ahead.
Yeah. Yeah. But it's been lonely for the first one to go do this with the agency. But now, I mean, the fact, you have the IgAN companies, you have companies like Travere really, sort of like being able to implement some new guidance in terms of approval. I think that's obviously just going to help. I mean, Vertex, as you mentioned, going to be taking APOL1 kidney disease to the agency, kind of helping solidify the regulatory pathway there. That Parasol group that I mentioned is actually taken on APOL1 kidney disease as their next analysis after they did FSGS, Luke. So it'll be interesting. They said they'll publish the findings this year, but that also can help establish sort of like the regulatory framework of what the right endpoint is for APOL1 kidney disease. I think that will be really helpful for all people working in the indication and disease.
Okay. So that's still, jury is still out a little bit on that one.
Yeah, they're supposed to publish this year, but I think I think you have a few things happening, right? You have Vertex kind of going into the agency, you have Parasol, you have our interaction. AstraZeneca also has a program in APOL1 kidney disease, so they're obviously going to be interacting. So I think next year, 27 will be a pretty big year for APOL1. I think in terms of not just drug development, also sort of like the interactions with the agency to give a little bit more clarity on what the regulatory pathway is. So I think big things coming with respect to better understanding the best way to develop new therapies for patients there.
Well, last thing I want to ask you, Jason, since you've been working on this company for some time, it's got a value of being high science driven and impact obsessed. We haven't really talked about that, but I know that's important, and you can measure that in several different ways. I mean, if you think out a few years, I mean, when most people think about genetic medicines, they think about rare disease and small patient populations, but you've got potential to serve small patient populations, but also large ones. How do you think about what your impact might be here at Maze 5 or 10 years out?
I mean, I think go back to like my own personal journey of like, how do we think about a career that kind of potentially can impact, you know, the greatest number of people and impact human health. And I look at kidney disease, Luke, as an opportunity to do that. There's so many people, little innovation, as kind of speaking about, and our goal is, with enough sort of like thoughtfulness and capability building, that we can eventually develop and hopefully launch these medicines for patients. I brought on some new people to our board. Hervé, who is the CEO of Insight, Neil Kumar, who I mentioned earlier, he's just joined my board, and partially because they've gone through that experience of actually not just developing but also thinking about how those things can go to patients, and so that is the aspiration, what we'd like to build in the long term. I think that would be, remarkable if a company like ours, starting off from scratch to actually get to that point, like a Bridge Bio, where they were able to, develop and commercialize multiple medicines. That's what we set out when, we were sitting in Charles Homsey's office, all those years ago. We said this is the kind of company we wanted to build. It takes a lot to get there. We know we have a long way to go from what we need to do here internally, as well as just sort of the company building. But that is what we want to do.
An enduring independent company that does the R&D and also the commercial side of the business. It brings together all of your experiences.
Yeah, no, absolutely. And I think that's why it was also important for me to bring people around the table in the company as well as on the board that have seen that before. Because I just having seen some of it, but then, making sure you have the right people around you. That was something I've always enjoyed in my career, having the right mentors and colleagues who can kind of help you think through, different things and difficult things. And I, benefited from that in my past. And obviously, I think that's important as we think about the next phase, this time next year, hopefully, in a pivotal study on APOL1, a couple other programs advancing through mid-stage development. But we have just like Bridge Bio, research efforts that allow us to think about applying our genetics-informed approach to other diseases. And hopefully we'll, be able to capture that ourselves or, as we've done in the past, think of creative ways either to externalize it through partnership.
Fascinating times. I'll be watching. Jason Coloma, thank you so much for joining me on The Long Run.
Yeah, thank you, Luke. Thanks for having me.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.